As Maria Paz told you, I will be speaking about my findings on the Atacama Desert, and I will be starting with this beautiful image of a Chastity Boston sleigh, in which how science actually imagined how Mars would look like in the 1950s. You can see even some green things, growing in the background. Now we know that Mars is a much harder planet. The atmosphere composed mostly of CO2 with some nitrogen and argon and some traces of other gases, which include water vapor, and also a very thin atmosphere, just 0.7% of what we have on Earth. As for temperature, we know that the temperatures on Mars, in the mean temperatures in a year go around minus 80 degrees, with the poles having the record of minus 120 or something like that. But in the summer and in the equator, in the day, temperature can reach up to 20 degrees Celsius. Although in that same day, summer day, temperatures at 9 will drop again to minus 70s. So this is a very extreme condition for life. We do know, although, that Mars was a wetter planet in its past, and that in time it got progressively drier and colder. So what are the chances of finding life on Mars now, with the focus on how much water is available now? We do know that we have all the morphological characteristics that point that there was abundant water on the surface of Mars in the past. We do know that we have all the geological evidences of a number of minerals like clays that only form when there is enough water for enough time for them to form. We have seen water ice that, thanks to the Felix mission, that it had just to scratch the surface of the pole in order to find water ice. And we do know also that in some days we can even have clouds formed by water ice. So water is there, and also even as water frost. And this is a beautiful image taken by the Viking mission, seeing how in the early hours of the morning you could see water frost there. Now, in order to study how potential life on Mars has adapted to an increasingly drier planet, we have to think that first, Mars is not just around the corner, it's quite far, that any given mission that you send to Mars is also quite expensive. For example, the Mars 2020 mission, which has now the Perseverance rover now in the surface, it costed more than $2.7 billion, which is more or less what the Mars Science Laboratory with the Curiosity rover also cost, about $2.5 billion. And now, even if you have the money, it's not just to get your own mission to Mars, because it's quite hard. In red, you can see all the missions that has been sent to Mars but has not reached it. And in blue, the one that has been successful. You can tell me that, yes, in time, we are getting better in order to get something on the first of Mars, but it is still not a done deal to get with something to Mars. So that's when you start thinking, well, how can I study the possibility of finding life in a planet as dry as Mars, but on Earth? And here you have the validity of the so-called analog model, places that have some of the environmental characteristics of Mars, which you can study. For example, this is the case of the Atacama Desert in northern Chile. Now, the advantage here is that for any given mission to the Atacama, I just spend $1,000 compared to the amount that I just told you in order to go for 4 days to the Atacama. So the Atacama is well known for being the driest place on Earth. And we know that because we have recorded many times that in the afternoon hours, no sensors are able to record no water in the atmosphere of the driest places of Atacama. I mean, I'm telling here that in a typically Atacama day, in about 2 or 3 p.m. in the afternoon, relative humidity drops to 0. What explains that Atacama is so dry? First, you have to think that all the masses of air coming from the Atlantic Ocean, when they start to rise up when they confront the Andes, they deposit all the water as rains in the eastern side of the Andes Mountains. So when they approach and then go over the Andes Mountains, they go down very dry. The so-called rain shadow effect, which happens here, but also many other mountain ranges in the world. But add to this that a similar phenomenon happen with all the masses of air that deposit the rains in the equator. Once they do that, they go up into the atmosphere and then they go down again at the precise latitude where Atacama is located. So you have an influx of very dry air coming also from above. And to also add that the Humboldt current coming from the South Pole and going north is quite cold. The waters are quite cold. So you have little evaporation in that area. And the few clouds that form there are efficiently stopped by the coastal range, which although is not as high as Atacama, it still causes a very effective barrier for those clouds not to go into the hyperarid core of the Atacama, which I'm showing here. So if you add those three conditions, you have a very dry place. And also consider that the Atacama is also the oldest desert on Earth. And that's because when the continental plate formed where the Atacama is located, it more or less stayed, as opposed to other places like India, for example, more or less in the same latitude. So it was always affected by the same environmental conditions for the last 150 million years, being an arid place, and for the last 15 million years, being an extremely arid place. And to add in the Atacama also, and this is a beautiful image of the ALMA Observatory in the Andes Mountains in front of the Atacama. If you measure the UV radiation that the Atacama typically receives in any given day, when you compare it with the non-UV index that tells you when to protect from UV radiation, in places like ALMA, for example, you can measure again in the afternoon hours with values of UV indices up to 20. So the UV radiation, UVA and UVB, are particularly strong in this area. So that's an additional factor of the extreme condition of the Atacama. In order to understand what this means for life, in particular microbial life, we have to think about water activity, which tells you on how bioavailable is water for life. Now, water activity is formally water activity is the partial vapor pressure of water in a solution divided by the standard state of the partial vapor pressure of water, which is the formula I'm showing there. It's more easy to understand that water activity can easily calculated by just dividing the relative humidity in any given environment and divided by 100. So that gives you a water activity chart in which you can put what we know of life on Earth in terms of the water activity available on how, and again, how water is available for life. We do know, for example, that water activity has for pure water is 1. Most bacteria will stop dividing at water activity 0.9. A few halophytic bacteria are still able to grow as water activity 0.7, and only a few yeasts are able to still reproduce at water activity 0.6, which explains why honey doesn't get contaminated, because it's so dry that nothing is able to live in there. So the question is, what happened at the ranges where the water activity I have typically measured in the Atacama? What happened with life at the water activity below 0.6? So to give just an example, this is the beautiful city of Strasbourg, where today you will have a humidity during the day of 74%, which translates to water activity of 0.74, and a UV of 3 out of 10 in the 1 to 10 scale, right? But compare that to the city of Pedro de Valdivia, which is located in the hyperarid core, the city where I was born. Today, the humidity will reach only 9%, equivalent to water activity of 0.09, and you did the extreme challenge you that is going off the scale of 1 to 10, right? So in the Atacama, all started with a study undertaken by the Jet Propulsion Laboratory, commissioned by NASA in the 1960s in preparation of the Viking missions, in which they described the microbiology of different deserts around the world. And this started in a now abandoned train station, which is close to the Atacama, to the coastal range, which is located here. Now, I know this place very well, and I know that that fogs are very common in this area. So although they found a number of microorganisms there in the 1960s, Actually, it is not among the driest places of Atacama, but that's where all it started. This story began, then continued almost 30 years later, when a team of scientists led by NASA came to Yungay, which is now is located in the high pelagic corridor of Atacama. And in there are the places which I told you that are dry as to get relative humidity of 0% in the afternoon hours. Now, why NASA came to this place? Because at that time they were trying to find a proper analog model of Mars. So they brought a number of equipment to this area, and they published in 2003, among others, bringing the gas exchange apparatus, similar to the one used by the Viking missions. And in 2003, they published in Science that they had found a place on Earth that could be the right limit for life, and also a Mars-like analog model. And they published that soil organic were at trace levels and only extremely low levels of cultivar bacteria. They could not recover any DNA from most of the soils analyzed. And when they repeated the incubation experiment undertaken by the Vikings, they showed the same active decomposition of organic species, which seemed to be commanded by non-biological processes. And they said, well, this is a good Mars analog model, and it seemed also that it is the driest place on Earth. Now, when NASA proposed its strategy to explore Mars in search for evidences of life, the first step they took was, well, let's see the evidence of where water was on Mars, and then the next step in order to see where we find evidences of life. So with a similar approach, I said, well, let's do the same thing, the same approach for the Atacama, and let's follow the water in the Atacama in order to see how life adapts to stream desiccation. And I started this in the coastal range, going inland in order to see how life adapted to that. So one of the first findings in the very front cliff of the coastal range, you can see that these black bandings here, those are epilitic biofilms. Those are a number of bacteria growing on top of the rocks. And here water activity varies between 0.7 and 0.9. That allows for life to grow exposed to the environment, because it's still receive enough water from the mostly from the ocean mist that cover that area. We have been able to detect members of the 3 domains of life here in these biofilms. We have been able also to grow a number of species of bacteria from that as well. And in that same area also, we have found another environment, which is our cave. This is a study with a NASA expedition in 2009, but which I continue later, in which we show that you could detect, in principle, the entrance to cave on Mars by infrared images. You could take an infrared picture of the area and say, well, given the temperature difference here, we can tell that this is a cave entrance. So I went into this cave armed with the hypothesis whether you could have even photosynthetic life well protected inside the cave. And actually at the very bottom of the cave, I found this beautiful green biofilm, mostly composed of a single species, where water activity again varied between 0.5 and 0.9, where light available for the photosynthesis was only 0.6%. And here I found a new type of microalgae that was able to live in those conditions. Now, the interesting thing is that the Sanidium species we found is closely related to other Sanidium species found in thermal environments. So we're still trying to figure out why we have this Sanidium in a non-thermal environment, in a non-acidic environment, as the cave wall I just showed you. In another cave of the coastal range, we found a different phenomena and another type of microalgae. And this type of microalgae, we didn't find it at the very bottom of the cave, but close to the cave entrance. And we noticed that that particular microalgae was growing only on top of the spiderweb that covered the cave walls, not in the cave wall itself, but only in the cave, in the spiderweb that covered the cave wall. And we learned that that was because here water activity now really drops dry, 0.1 to 0.9. It's still high during the night, but it's really low during the day, because it's still close enough to the coast for these local folk to go into the cave. So we learned that this microalgae are able to live there because the silk of the spider web is quite hygroscopic. So we went back at 3 a.m. in the morning, and we found this to be a new species of Duraliella, which is actually, you can see the aquatic members of the Duraliella when you go into the San Francisco airport, because they are grown in order to get carotenoid. On another place of the coastal range, as I said, this is commanded by the fogs that cover there, and we come back to the 3 a.m. and we could see the spider web covered by little drops of water, confirming our hypothesis that we're getting water from the fog water. Why all these findings are interesting in the case of Mars? But we know that are caves on Mars. Probably this is a skylight of a lava tube, and so we think that as Mars get drier, maybe life found refuge, a place of where a protected place like caves, far enough for cave entrances, so not to be affected by UV light, but close enough to get enough light for photosynthesis in order to grow there. So you may not have now life, but maybe you have the remnant of that in the very ancient cave of Mars. And we know that in the case of Earth, life has invented different type of chlorophyll in order to use different type of wavelength in order to use the visible light, even to the infrared, in order to use all the cave environment. And it could be also the case which happened on Mars. The next step is to explore Mars. We are now, in general, I speak devising the how you would explore cave on Mars, which will not be an easy task. You have to develop new technologies in order to go in there and explore. And some of them are already being developed in terms of in artificial intelligence applied to robots that can go inside those caves and take the proper samples and then communicate all the findings to the surface. Some of these caves are can be quite big. Here you can see a few persons sitting inside this cave. So people is already thinking that maybe also, in addition, caves could be our first home on Mars. And for me, it's mind-boggling thinking that we're doing somehow retracing the first step of our ancient relative. As we did on Earth, we're doing the same thing on Mars, right? Now, if we go in and into the coastal range, we find a different phenomenon. Here, fog are still the main source of water. And in a few places of the coastal range, when you go into the hills of the coastal range, you'll get to see a very interesting phenomena in where you get to see these quartz outcrops. And when you pick up 8 out of 10 of these quartz rocks, you will find this beautiful green biofilm. These are hypolytic biofilm growing under the rocks, in which they are able to use this protected environment in order to colonize and grow it. Now, the species present here, again, are quite complex. Here, water activity, again, is quite low during the day, although it can be quite high during the night. These biofilms are composed by a number of different species, which you can even find attached to the soil where the quartz is located. That's because the photosynthetic active radiation between 400 and 800 nanometers is able to go across the quartz, so they can be used for photosynthesis. And also because the quartz stones, because its physical nature, it remains colder than the environment. So during the night, all the surfaces are covered by the condensing fogs. All the water is condensing in all surfaces, but during the day, since the quartz rock is colder than the environment, it can keep the water for a longer time during the day, thus allowing for the colonization of a species like cyanobacteria, which, in addition, the cyanobacteria are known for being high producers of these exopolymeric substances, which surround all the community, which are quite hygroscopic, so making even the environment more amenable for water condensation and thus colonization. Here we have found, again, members of the 3 domains of life, archaea, bacteria, algae, and under any given quartz, you will find up to 70 different species. We have been able to grow these communities in the lab. We have isolated a few species like Leucopsopsis, a new type of bacteria, and start asking the molecular question on how they are able to survive water activities during the day of just 0.16. In this area as well, you can find the colonization of gypsum. This is a surface gypsum. This is the way you can see it on the surface, but if you take one of these and crack them, you will see this beautiful green layer here, mainly composed again by different type of algae and cyanobacteria. The water activity in these gypsums is more or less the same as I just showed you for the quartzes. So life is also available to use that kind of environment, which in the case of Mali is interesting because it tells you the importance of microhabitats as island of diversity for life. It called my attention that one of the first pictures that taken by Perseverance was this very interesting rock covered by little holes. And this is the type of environment I have seen in Atacama, where it makes the whole difference from microbial life. If you have a protected environment, for example, that can protect you from UV, that with the with the Fox event, they can condense water inside, or the very rare rain event, they can condense water, as I have seen in the driest side of Atacama, telling you about water getting trapped there and being evaporated since who knows what. That makes the whole difference for life. And actually, I have seen the same phenomenon again in the Andes Mountain, where you find a whole microcosm of different types of bacteria, microorganisms. They are living in this little pockets of water. Same thing with quartzes. I just told you the hypolytic phenomena, but you can see all of the cosmoendolitic phenomena where cyanobacteria are able to grow in the cracks of the main outcrops of quartz itself. So I just wonder why NASA didn't get one of these lasers into the caves in these little caves in order to see what they could detect there, being such a nice protected environment. Now, if you go inland into the hyperbaric core, then you go to places like Yangai, which NASA claimed that they were the Driest of the Atacama. But as Maria Paz told you, I was born and raised in Atacama, and I knew there were much drier places than that. I knew there were places were much drier than Yangai. Even though the water activity between Yangai now drops to 0 in the afternoon hours, as I told you, but it can get still up to 0.8. And that explains, for example, that in Yangai, for example, if you crack one of these, this is the area was first explored by NASA, but if you explore the rocks are in the front of the picture here, those are halides. And halides are composed of mainly sodium chloride, that's the common salt. And this gray banding you can see here are cyanobacteria that I just highlighting here that are able to live inside those halides, telling you that independent of the source of water, that can be high relative humidity or focal section, etc. There's a number of mechanisms that explain how you can get water into those halides and then get those cyanobacteria to be active. There is enough water for that. So in 2015, I reported what is truly the driest place of Atacama, and thus the driest place of Mars, which is Maria Elena South, in which water activity at best can get to 0.5. Now, knowing that this is the one was supposedly the one of the driest places on Earth, we go into the subsurface in order to see how water activity changed there. You can see this the surface profile where you get relative humidity. I'm sorry, in this case, you can get high relative humidity during the night, then very low during the day. That's the very normal, common, typical cycle in Atacama. But look what happened at 5 centimeters of depth, 20 centimeters, 50 centimeters, and 80 centimeters. When you go well enough into the surface subsoil, water activity remains constant in time as low as 0.7. So we thought that maybe we had get a place as dry Mars, and actually it was the case. When we compare those days with the same measurement that were measured in the Gale crater at that time, we matched the lowest values of relative humidity measured on Mars. And for the first time, we saw, well, maybe we have reached the so-called dry limit for life on Earth. How we reach a place so dry was nothing was able to survive in it. But to our surprise, when and taking all the precautions for it, we inspected the soil in depth. We found a number of different microorganisms just from seed genesis: Streptomyces, Bacillus, and Eudematophilus, which is the black species here, which is very interesting because it's able to survive from the reduction of manganese. So they could be the primary producer, telling you that even in such dry condition, life is able to prosper. Now, the next question was to ask, well, how life get to these places? And for this, we use this very technological instrument, which we call the portable wind vector assessment tool, in order to say, well, maybe wind is able to transport life from places to places in the Atacama. And knowing that in any typical day, wind will start to pick up speed in the afternoon hours and then go inland, you say, well, maybe this is the way life is coming from the ocean and getting to colonize the places inland. And for that, we set different wind transects. We set different gross media in plates across this transect. We were able to match the wind speed and tell, yes, we do have a stronger wind speed in the afternoon compared to the morning. We were able to trap dust, and from that dust, although just a few milligrams per plate, we were able to find all that type of microbial life being supported by the wind, and identify the species of where you would find it in particular site of each transect. And with the interesting finding that when we modeled the wind fluxes, we could see that most of the transport process took place during the night hours. And then most of the local transport took during the day. So it was during the night that life was able to traverse the whole Atacama in just a few hours. So being a very efficient transport process. So what tells us in the case of Mars? Well, we do know that Mars is affected by, in this case, global scale wind dust storms. So it may have happened in the past on Mars that could be an efficient mechanism for transport places where you will have life to other more stressed places. And also, even now, if it is true that in some of the craters, water is being released in its wall, it may be the case that if the transport takes place during the night, you could take microbial life on Mars from protected place to other protected place, either take place on Mars, as is the case in the Atacama. And it's also an important lesson for planetary protection, but we do know that as soon as you get man on Mars, it will be impossible to get protected the planet from this very same process in order to disperse microbial life all across the red planet. Now, changes came again in 2015 when we start seeing these very rare lagoons. We saw that they were man-made in 2015, so we just continued our way that day. But what is going on here is that, let me explain first that it does rain in Atacama, but only in the foothill of the Andes, when in what we call the Andean winter, which by the way takes place in summer. We do have some rains in the foothill of the Andes here at the other side where the Atacama is located. But to be truthful, the environment in the foothill of the Andes is quite different from the environment that I'm showing here in the high parallel core. So that there's a very different ecosystem. This is the foothills, the high parallel core that I have been showing you. But what happened in 2017 is that the temperatures of the Pacific due to global warming were abnormally high, so great masses of cloud came from the other side and went inland and causing this lagoon that had never been seen in these areas. So we saw that life in these areas would be like welcome like manna from heaven, which actually is the case in the southern edge of Atacama, which is located 600 kilometers south from the true high parallel core, more closer to the central part of Chile. But here, we found that in this water of this lagoon, we could only find 4 species. Coincidentally, with the 4 morphological types we could find in the water, and actually we were able to grab one of the species also. And the question is that when we compare those 4 species that we could find in the water and compare it with all the species that have been found before in the soils of Yungay, we found that we couldn't find any of the 85% of the species were not already were in those lagoons. And this suggested that what was going on here, that these species are so well adapted to the trap and the efficiency use of water. When you just put a lot of water in a short time of it, they die from osmotic shock. And why we're seeing that happen, it first tells you on the extreme adaptation of the species to the lack of water. And we know that is the case because we could see the biosignatures left behind by the active species that we know that were there, but also we could see the signature of the degradation products of like pristine and phytin that come from chlorophyll, which is typically from the cyanobacteria that I just told you about, which were present in the halides that were dissolved in this water. And actually tell you also a very interesting lesson that thinking about the biochemicals and that in two of this experiment you added water, maybe you reproduce what you just saw in the lagoons where you add water to a sample, you degrade any given cell that could have been there. And if you add the typically very highly oxidized species of the margin soil, which you also find in Atacama, which like percolate, you may have annihilated any type of organic before you even detected it. So it's a very important lesson, and actually we are trying this experiment right now. Change things a lot when you don't add as much water as having a lagoon, because we found that if you dig into Yangai now in the soils of 2017, you will be able to see a clay layer made mainly by smectite, and it would, in this case, was completely opposite to the case of Maria Elena. You remember that all the values as we went deep were low. Here was the opposite, because of this clay layer, you have a water activity of 0.7. It was trapping water. We still not sure what the water source, but it was wet enough in order to sustain life. We could find again a member of the Xeromine of life, including a number of different types of bacteria and also archaea, which are the species that are in gray here. And we were able to grow a number of other species as well, finding the type of vegetable you would expect to find in haline soil like this, like Halobacillus, which tell you that if you have enough water for life, you would have it in the subsurface, as it is the case, with a very extreme environment just a few centimeters above you. So as you can see, and I'm close to finish here, if you approach the Atacama from the closest side to the coastal, to the ocean, the Pacific Ocean, you will find that life is able to grow, expose to the environment, as I told you, because water activity is quite high yet in order to allow the epiphytic mode of life. If you go inland into coastal ranges a few kilometers, you will get to see an immense drop in water activity just to 0.1 during the day. So life has to find protected environment, like going into quartz rocks, under quartz rocks. But if you go like places to Yangai, for example, you will get to see that life again have to find protected environment living inside halite, where water activity can easily vary between 0 and 0.8. But if you go to the truly dry places of Atacama, like Maria Elena, you have to go underground in order to survive with water activity or just 0.1. So another way to say that is that same process may have happened in the evolution of life on Mars, where you could have life exposed to the environment in the epiphytic mode way in the first days, and as progressively Mars get drier, you would find more protected places. Another way to say that in the case of Atacama, tell me what age of Mars, epoch of Mars you are interested in, and you can tell you exactly where to go in order to understand how life has adapted to that particular conditions, which validate the missions that are thinking about drilling into the subsoil in order to look for evidence of life. You could have, you could have a very extreme environment on the surface, but if you just dig, as we did in the Atacama, just 30 centimeters down, you will have evidence of life. So to answer the question, is Mars too dry for life as we know it? Considering what I have seen in the real world, and with that I mean that it's not through modeling or something like that, I have seen that if you find protected environment like quartzes or caves and gypsum, or in the underground where you have sources of water that you cannot easily see or detect from the surfaces, and even if you go to really dry places to Maria Elena, whether you are at the limit of detecting water, you do find life. So is Mars too dry for life as we know it? My answer is no. For what I have seen in the Atacama, I think the true question that we should hit in the case of Mars is whether life ever originated on Mars? That's the true question to follow, because if that happened, it should have adapted as it did in Atacama. And actually, this is a very interesting question, a very complex question. There's just a few works on trying to figure out whether life ever originated on Mars. It is a complex question here to solve on Earth. It's even more complex in order to solve it on Mars. And with that, I end, and I open to any question you may have. Thank you very much.